Immunology, cells of immune system. Cells of immune system. There are various cells that are there in the body, blood cells and other T cells, B cells, which help in the immunity of the body. Hematopoiesis. All blood cells arise from hematopoietic stem cells. They are scarce, difficult to grow in vitro, can isolate using monoclonal antibodies and flow cytometer. Early in hematopoiesis, multipotent stem cells differentiate into two pathways, either lymphoidal progenitor or myeloid progenitor. Stem cells are derived from pluripotent stem cells of bone marrow. They follow either of the two cell lineage, myeloid cell lineage or lymphoidal cell lineage. So this diagram shows the myeloid cell lineage and lymphoidal cell lineage. There are two different types of immune responses: non-specific and specific immune response. In myeloid, it is non-specific, that has been brought out by monocytes, macrophages, granulocytes. T lymphocytes, B lymphocytes, larger granular lymphocytes are specific immune responses that are brought out by lymphoidal cells, cells of immune system. Now we'll see the cells of the immune system. First is monocytes. Monocytes are mononuclear, that is horseshoe-shaped nucleus is present. They are phagocytic in nature. 4 to 10% of nucleated cells in the blood. They migrate to the tissues and become macrophages. Eosinophilic lysosomal granules, they actively participate in phagocytosis as wandering phagocytes. They are activated by various stimuli, are gamma interferons, interleukins, and some components of bacterial cell wall. Activated macrophages are referred to as antigen presenting cells. Their activated macrophages secretes factors like tumor necrosis factor, that is TNF, which can kill tumor cells. And this is the activated macrophage and macrophage. Granulocytes, so called because of the granule in the cytoplasm. Cells have cytoplasmic differences with unique staining properties. And based on the staining, there are three types of granulocytes: neutrophils, basophils, and eosinophils. Granulated neutrophils. They are multi-lobed light granules. Granules contains enzymes of phagocytic activation predominant at the site of the inflammation. Some substances secreted during inflammatory reactions acts as a chemotactic factors that accumulates neutrophils at the site of the inflammations. Highest number of polymorphonuclear leukocytes is the indication of infections. So when there is more number of neutrophils, it shows the infections. Granulocytes, they are phagocytic in nature. They are phagocytic in nature, generate antimicrobial agents. Neutrophils are very short-lived when compared to macrophages. And this is the neutrophil diagram. Granulocytes. Next granulocytes is basophils. They are lobed Nucleus heavily granulated. Granules contain histamine and other vasoactive substances. They are non-phagocytic, have receptors for unique class of immunoglobulin IgE mediated allergic reactions. This is the structure of eosinophil. Lymphocytes. 20% of leukocytes are lymphocytes. Two distinct types of lymphocytes are smaller lymphocyte, which have higher nuclear cytoplasmic ratio, larger lymphocytes which are granulated with low nuclear cytoplasmic ratio. Some smaller lymphocytes are granulated and classified into T lymphocytes and B lymphocytes. Those that have not interacted with antigen are called naive cells, and interactions with antigen, they get proliferated into two different cells, effector cells and memory cells. Memory cells are stored and effector cells are taking part in the destruction processes. Antigen presentation. Helper T cells. T cells, when get activates the suppressors, T cell suppresses activates, which shuts down the both cell and humoral immune response. When T cells bind to B cell to antigen complex, it leads to the formation of B cells to get multiplied and leads to humoral immune response. When T cells produces MHI factors, macrophages congregates at the site of the infection. When T cells produce interleukin, they take along with the multiplications of T cells, they get involved in cell-mediated immune response. When T cells activates the induces, it initiates the difference of T cells. T cytotoxic cells. CD18 glycoprotein, antigen receptors on the surface, binds to the fragments of antigen displayed by the class 1 MHC. Recognition of MHC antigen complex initiates the differentiation into effector cells called cytotoxic T lymphocytes. Once the activated T cells release cytotoxic perforin and granulysin, which form spores on the target cell plasma membrane and leads to apoptosis, which is also called as programmed cell death. Activated T cytotoxic cells expresses surface cytokines Fas-ligand that can bind to Fas molecules on the target cells. So Fas ligand interaction forms the route to dispose T lymphocytes during the development. B lymphocytes, they are B cells, they are very important in antibody mediated immunity, display membrane bound immunoglobulin, that is Ig, which is an antibody, secrete specific antibodies in response to antigen stimulus. Their site of maturation is bursa fabricius in case of birds, and in case of mammals it is bone marrow. Larger, there are B lymphocytes, there are two types of subsets, T cell independent cells, T cell dependent cells. Once antigen is encountered, differentiation, plasma cells gets differentiated into antibodies that secreted die within 1 to 2 days. And the memory cells have same membrane bound antibody as parent B cell, have longer lifespan. T and B cell. Thank you.